Hi, everyone. Thank you for introduction. Thank you for giving me this opportunity to present my research. So today, I would like to talk about how we can use deep learning for atomically resolved imaging techniques, specifically for scanning transmission electron microscope and scanning probe microscopies. I will mostly talk about electron microscopy. All right, so when it comes to applications of advanced data analysis or AI to the field of nanoscience and nanotechnology, then the way I see things right now is that we have actually two different universes. Now, we have universe one, where we use experimental and theoretical tools to study materials at the level of individual atoms. And here we try to understand how materials behave, physical and chemical processes at the atomic level, and we try to understand them and to use this knowledge to learn how to control them in order to enable atom-by-atom fabrication of materials with desired functionalities, which is the ultimate goal of nanotechnology. But we have a problem here. It's a huge problem. And the problem is that we are getting much more data than we can analyze. So right now, sometimes a single experiment can give you gigabytes to terabytes of data, and you do these experiments on a weekly basis. So it will, using the common tools, kind of manual analysis that people usually in this domain do, it will take you probably a year to analyze the output of a single experiment. And again, you do those experiments on a weekly basis. Well, luckily for us, there is this second universe where people have already figured out how to work with these large amounts of data, how to do data mining on this type of data. So this is the universe where you have all these fascinating developments like self-driving cars, which use a lot of image and video data analytics, advanced facial recognitions, unsupervised translations, and so on. So what we are working on at IFM, which is Institute for Functional Imaging of Materials at the Oak Ridge National Lab, is that we are trying to bridge these two universes, open, let's say, a wormhole, and keep it open long enough to make some practical, to get some practical results, okay? So let me explain what I mean just in a few examples. So just take a look at this image. This is an electron microscopy image of a two-dimensional material called graphene. So graphene is a sp2, you have sp2 bonded carbon atoms arranged in a honeycomb-like structure. And I think I have 6 consecutive frames here in the loop. And so what you see here, first of all, you see graphene lattice. You don't actually see atoms, you see these hexagons, but as a domain expert, I can tell you that there are atoms in the vertices of these hexagons. Now, this lattice gets reconstructed in some areas, so you don't see hexagons anymore, you see some 5-7 and 5-8 member rings. Now, in addition to that, you see formation of some holes, multiple atoms are missing, there these holes get healed, they have some peculiarities in their edge shapes, and then you see some impurities jumping around these holes. So, and everything is happening under the electron beam irradiation. So there is lots of very interesting physical and chemical processes that are happening here. And the question is, how do we extract all this relevant information? And to do this, we need to map the positions of all the atoms here, all the impurities, analyze the reconstruction of the edge of the hole, and it turns out that the most of the current methods do not allow you to do it fast enough, and definitely not in a real time. Okay, the second thing I wanted to talk today about is that, second example I wanted to give is that, so the modern day microscopes allow you not only to do imaging, but you can also use them to manipulate matter at the atomic level. So you can use them to move atoms or to change structural state of molecules. You can do it either with a tip of scanning probe microscope, or you can use electron beam to push atoms or to pull them apart. But right now, all this is done by a human operator. Everything is relying on the educated guess of a human operator. The reproducibility is not that great. Efficiency is very slow. So it's now, you can spend a couple of months to, put these few atoms together, publish a nice paper, but you cannot turn it into a tool. So what is really needed here is that we want to have an image-based and physics-based feedback for atomic level manipulation of materials structure. All right, so in case what I said here was very confusing. I put everything together on just one slide, okay? So this is the way I see this area. So we always start with, we want to have a rapid physics-based image recognition tool. And from here, we will learn a lot of new interesting physics and chemistry on single atom and single atomic defect level. So this is where we will discover a lot of new science. But then also this will serve as a critical part of what I call a self-driving microscope. So this image-based analytics goes here. Then you will combine it with reinforcement learning on molecular dynamic simulation to learn optimal control policy. And then this will all lead one day to a controlled atomic scale fabrication, which is, again, the ultimate goal of nanotechnology. So today, we'll mostly talk about this part. OK, now I think I don't need to introduce what deep learning is for this audience. But basically, the way I usually like to explain it in very simple terms is that you have input, you have output, you have neural network, and then in between them, and then you match input to output by adjusting weights. And the difference between deep network and shallow one is that the deep one has more units and layers, and the deeper network becomes, the more capable it is of capturing abstract high-level features and not being very sensitive to pixel-level details, which seems pretty important for our task. And but then the question is, OK, so we read every day about applications of deep learning to macroscopic world. How about the world of atoms and molecules? And I like to argue that actually physics and chemistry makes our life much easier here. So this is one of the way to study atomically resolved structures that I came up with. I call it weakly supervised learning. And the idea here is that you start with a very limited number of classes, because this is usually all that you know in the very beginning. Say, for example, if you're an experimentalist and someone brings you a sample, and all that you know, well, they tell you the name of the sample, so you know what type of lattice you have. Is it square, or honeycomb, and so on? So this is one class, your lattice. Then people like to dope their samples with something to tailor their functionalities, so you know what type of dopant you have. And then just from your general background, you know that there is no such thing as an ideal periodic lattice. There are some missing atoms. OK, you have vacancy. So these are the three types of three classes that all the materials have. So you train your model using theoretical simulations based on with a physics-based algorithm, such as multislice for electron microscopy. So you train your model, and then you take your experimental data, and you apply your model to the experimental data. And then what you typically see is that in addition to just these three classes, you will have classes that can be a combination of, for example, two classes, like a dopant can sit on the edge of vacancy, or one class can split into multiple classes. Like, you can have dopant with a three-fold coordination with a nearby lattice host, or four-fold coordination. So what you do is that, and I will talk more about it in details later, but what you do is that you map the pixel level output of your deep learning model onto the graph structure, and you introduce specific chemical constraints here on the maximum and minimum bond lengths, maximum number of connections that atom of specific type can have. And in here, you make a transition from pixel level based analysis to analysis based on physics and chemistry. So analysis based on the length of chemical bonds, bond angles, a coordination number, and so on. And then here you will typically end with having like dozens of new defect classes, and then you can plug them into the simulations, and then you will update your training set, and then instead of 3 classes, you will have like 20 or 30 classes. So this works. OK. Yeah. Sorry. OK. So oops, sorry, I went too far. And OK. The type of deep learning model that we use is this segment-like structure, encoder-decoder type of architecture. So it consists only of convolutional layers. You don't have any fully connected layers here. And it returns you a pixel-wise classification maps, so a probability of each pixel being a certain type of atom or defect. Okay, so this is your input that for electron microscopy data, you get it from simulating how electrons scatter from atomic nucleus. And so this is your ground truth. And so you don't simulate all the training set with this multi-slice algorithm. You actually simulate dozens of images, and then you do data augmentation. And you don't do it randomly. You actually do it to account for certain instrumental factors as well as uncertainty in your model. Like, for example, when you put sample in a sample holder in an experiment, it doesn't always stay in the same way. It can be rotated. So you take all the rotations with respect to the lattice symmetry into account. Then in the experiment, you zoom in and you zoom out. So you want to take this into account. So you do this augmentation. Then you apply global and local distortion to the lattice. And then, of course, you add noise and blurring in the range compared to the levels you see in the experiment. And so you get from dozen of image to like 1,000 or 2,000 or 3,000, depending on how you do it. And then you use it to train your model. I always forget that I can actually turn to the next slide, this one. So I always go back to my laptop. OK, so let's now see how it actually performs on the experimental data. So this is the really cool thing that I'm very excited about. We trained it only on theoretical data. It was simulated data with using the known physics, OK? And then we apply it to experimental data. This is the image I showed you on the second slide. And actually, so here the atoms do not produce local maxima. All you see are these hexagons and heptagons. And actually, it was able to find all the atoms. And the interesting thing is that, so this is the softmax layer of a deep learning model. And the interesting thing is that, so I trained it only on hexagonal lattice, but it was actually able to generalize well enough to find these peculiar non-hexagonal reconstructions, which actually are pretty important in this material. And it was also able to find vacancies and impurities, which is great. Well, I tested it just with, by artificial increasing level of noise to make sure it doesn't make a magic and produce atoms out of nothing. OK, so now we take this input and we map it onto a graph structure with specific, and we introduce some specific chemical constraints when we do this. And now we can actually make a transition from this one to this one. And by doing so, we can now categorize these atomic and defect structures based on their chemistry. And then we are, and it takes basically, you get from here to here, on this laptop, it will take you a second, which is pretty cool. So you can now analyze lots of data. And here is just an example of what if you apply it to the very short movie with just 4 frames, but you can already discover a lot of new things. Like in here, you start with this 4-folded silicon impurity, then one carbon atom gets knocked out. You have vacancy here that you can actually recognize, and then vacancy and impurity switch their positions, and then it captures another silicon impurity and makes this very peculiar structure. And we were actually the first one who were able to identify such kind of structure in graphene. So again, we started with just 3 classes, and now we can have much more. And then we can use them to update our training set and make a model with 20 or 30 different classes. OK, so one thing I like about machine learning in general and deep learning in particular is its ability to generalize beyond the training set. So here, I took a model trained on graphene and applied it to a different material with a different, somewhat different lattice. It's molybdenum diselenide doped with tungsten. And it was able to find atoms and defects in this material as well, although it was never trained even on the simulated data on this material. And here, so here, this is the lattice. So, you have 2 inequivalent sides here. You have molybdenum atom in one side and a stack of 2 selenium atom in other side. And so it means that you can have half vacancy when only 1 selenium atom is missing and full vacancy when both selenium atoms are missing. And so actually, we were able to distinguish between the full vacancies and half vacancies, which is pretty cool, and then also find dopants. So this ability to generalize is quite wonderful. OK, let me skip this slide. Here I just wanted to show it can be applied to other microscopy techniques as well. OK, so the next problem that we are interested in. So we are very interested in studying electron beam matter interaction. Okay? And so the experimental data that we get is something like this. This is a 2-dimensional tungsten disulfide material under E-beam interaction. And so you can see irradiation. And so you can see that some point defects start to form. Then they move, they merge into larger defects. So you see emergence of new phases. And what we want to have, so this is what we have. And what we want to get is something like this, where we see how different type of defects evolve in time. So basically how we get from here to here. Okay, so this is the, well, how we'll do it. Okay, so what we did is first we need to find defects, and we don't have any theory about what type of structures we have there. So we cannot just simulate the data. and train a model and simulate data. So what we did is that we realized that actually, well, every defect is associated with a violation of ideal periodicity of the lattice. And so what we did is that we took a first frame, or you can take a frame just before recording this movie, and you do some simple Fourier-based analysis on this image when the global periodicity is still maintained. And you can map positions of all the point defects. Now, you do data augmentation, and then you basically, this is your input, and this is your ground truth, and you train a model to find these defects that break lattice periodicity. And then once you extract enough, and then you apply it to the rest of the data, and then you do some Gaussian mixture model based on mixing. So let me show you how it works. So how it works. So we trained it using just a single frame, and then we applied it to the rest of the movie. It was able to generalize well enough to find previously unseen defects in the rest of the movie, and it is stable even with respect to formation of these larger defects. And then we performed classification using Gaussian mixture model. So we mixed them into the physically meaningful components, like 2 different types of vacancies, substitutional dropant, datum, and so on. And then we reconstructed these trajectories. So this is basically time, it's a number of frames, this is x, y. And so you can actually see that there are different statistical behaviors here, different diffusion behavior, and so on. So this is pretty important because even if you have an army of graduate students, there is no way you can actually, reconstruct this trajectories. So that's very important from the domain science point of view. I think we were the first one who actually were able to do it. And now, let's pick up one of these defects and analyze its, let's say, diffusion behavior. Right? You can extract its diffusion coefficient within 2D random walk approximation. And what else can we do? We can analyze local distortions for the specific type of defects, or you can actually see that some of them are switching along their trajectories. And what if we analyze it as a Markov process, and then we can reconstruct the transition matrix with the transition probabilities. So the important point here is that we started with this, grayscale movie with like hundreds of frames, and then we were able to get from this movie to this physics and chemistry. And this is how I see, we use advanced image analytics in material science. We use this as an accelerator for scientific discovery, okay? I don't want deep learning to do scientific discovery for me. I want it to be this engineering tool that will accelerate dramatically scientific discovery. In this case, we were able to analyze these trajectories that there is no way you can do it with the standard tools that people use in this domain. How am I doing on time? How am I? Okay, so I have like 13 moments. Okay, so 13 in total, but then there is time for questions. Okay. How many? Okay. Okay, all right, okay, thank you. That's important distinction. Okay, so another thing that we are interested in is mapping atomic scale potential energy landscape from analyzing atomic dynamics. So basically, the type of experimental data that you have is this one. It's very noisy one. We need to do imaging very fast to capture these dynamics. And so we want to reconstruct all the trajectories and then use it to map potential energy landscape. But the first thing is we cannot actually do it directly on this data, so we need to clean it. And I found that one way to do it is using this so-called denoising autoencoder, specifically deep convolutional denoising autoencoder, which you teach it. Again, I used theoretical data to teach it what noise is, and it can remove all the noise. Depends on how you define the noise. And it also can reconstruct missing parts of your data. And so then it works really perfectly for this type of data. This is the original experimental data, and this is the output that it gets you. So you have 2 types of atoms moving here. It removes everything else and returns you basically these 2 classes of atoms on the uniform background. And then it's pretty straightforward to map their trajectories from here. So you can reconstruct their motion, this one-dimensional representation as a function of boundary index. And then you use this information to actually reconstruct atomic potential. So again, at the step age of graphene. So again, we started with this noisy movie, which didn't seem like much interest, and then we actually arrived to this atomic scale potential. So from image to physics via deep learning. So I think that's another example of how it can accelerate a scientific discovery. Okay, one other thing that I wanted to show today is not directly related to atomic scale imaging, but it is related to the type of problem that we are solving with microscopes as well. So here, what we see is that, so the type of data that we have are the atomic coordinates and how they change as a function of temperature. So it's atomic coordinates of a material called relaxer ferroelectric. This is a very interesting material with peculiar physical and chemical properties. It is characterized by very high electrorestriction, so it's macroscopic property, but people studied it for 50 years and they still have no idea what is the microscopic mechanism behind this macroscopic phenomena. But generally the idea is that at the high temperature it exists in the non-polar paraelectric phase. Then as you cool the system down, the polar domain starts to form, but they are oriented in the different directions and they move. And then as you cool the temperature further down, they kind of freeze. But can we actually, learn about it in the unsupervised function? Can we extract some parameter that will approximate an actual physical order parameter of this system? And so we used a tool called deep variational autoencoder. It's actually called variational autoencoder, but I always put this word deep. It sells better. So basically, you have unlike regular autoencoder that learns arbitrary function that maps input to output, this one actually learns probability distribution modeling the data. And so now I use the, you can use convolutional layers here. I just flattened it and used the fully connected layers, because it worked. And basically I chose only one latent parameter, so you compress it to one lattice parameter, and then so then this is this lattice parameter. So latent parameter is a function of temperature, and this reconstruction loss is a function of temperature. And this is one of the possible theoretical distributions of the system with a physical order parameter. So you can see that we actually were able to find a transition-like behavior at 230 Kelvin, which is, I'm sorry, it's actually, this is 230 Kelvin here, I'm sorry, it's normalized. And then you can see that below this transition temperature, you see some oscillations, and we also were able to find these oscillations. So it's pretty cool that we can actually find the exact temperature of transition and also in general reproduce this spin glass-like behavior below the transition temperature. Okay, so I wanted to just spend 2 slides to tell you what are future challenges in my opinion. So I think that one thing that is kind of troubling right now is that we collect a lot of data, we analyze a lot of data, but how do we actually store it? Well, there is no proper way of doing it in this particular domain. So, part of the data is stored on, on my desktop. Part of the data is stored on the desktop of the operator of electron microscope and so on. So, one thing that we kind of need to do is create this database of atomic defects and their functionalities. I call it, materials ImageNet, in the sense that the way I want to use it is that, you create this database, and then just like ImageNet and similar database are used to train state-of-the-art deep learning models or machine learning models for natural image recognition. Here we can use this database to train machine learning, state-of-the-art machine learning models for atomically resolved imaging. Because right now such thing doesn't exist. And it can be complemented with theoretical calculations. Okay, so here are some details. It's kind of Oak Ridge specific. So, and another thing, as I mentioned to you, is that, all right, so what we have now is we have deep learning tools that allow you very fast, very accurate and physics-based identification of atomic scale object in your images. Okay, let's take it to the next level and actually make a self-driving microscope. How do we do it? Well, the one thing that we need to do is that, okay, it sees something and then what does it do next? So we want to learn an optimal control policy for each specific type of material, for each specific type of system. One way to do it is to use reinforcement learning on molecular dynamic simulation to construct library of cause and effects rules. And then, you recognize something, you search this library, and you make a decision and implement it via the feedback loop. and that's how you get to fully autonomous microscope. So basically, the idea here is that just like you teach a robot to kick a soccer ball into net, so here your robot is your tip of your microscope or your electron beam, and your ball is an atom that you want to move, and your net is positioned in the atomic lattice to where you want to move your atom. So, okay, and with this, I think that's all I want to talk about. Thank you very much for your attention.
So we have a little bit of time for questions.
Yeah, my questions relate to like the input. Can you use the microphone to the other? My questions relate to the. I'm wondering about the size of the input image that the neural network actually sees, and how sensitive the analysis is to that resolution, and how you choose that. That's a great question. Yeah. So in principle, since we are using fully convolutional networks, you can actually do it with a variable input size. That being said, because of the let's just say people who collected this data were not really, there was no specific, methodics for how they choose their resolution. I mean, in a sense that you can get one image with a 128 by 128 resolution, and another image with 4096 by 4096 resolution. So it was a little bit tough to actually, train. Well, I mean, I think it's possible to actually, make a model that will do recognition of both of these images. So I settled down to it's 256 by 256 pixels. So because it so we're interested in learning these bond lengths and bond angles. So for that purposes, we tested it on the known defects. It works well, and so this is enough pixel resolution for this type of problem that we're dealing with. That being said, yes, that is something that we need to. It works for specific type of problems that we are working on, but there will be some images potentially that will, the model will fail to analyze, let's say too large or too small. Actually, too small is not a big problem, you can do it manually, but too large, right? So it's a good question. If you do it kind of patch by patch, it will create these extra atoms probably at the boundaries. It won't be very super accurate, and you may confuse those boundary effects for some physical phenomena actually, right? So yeah, so I always give long answers to short questions, but the question is, so we used for the type of data that I showed you, we used 256 by 256. If image was larger than that, I downsampled it.
Great. So let's thank the speaker.